All right, and welcome back. Today we'll be taking a look at our third of the macromolecule groups, nucleic acids. All right, so starting off, there are two types of nucleic acids. There's the very famous deoxyribonucleic acid, otherwise known as DNA, and then the not quite as famous, but still very famous, ribonucleic acid, known as RNA. First thing we'll take a look at here are the structure of the monomer units of nucleic acids, and those are called nucleotides. All nucleotides are made up of three parts. We have a phosphate group, a pentose sugar, that means a 5-carbon sugar, so pentose for 5, and a nitrogenous base. You may have before have heard of DNA and RNA and thought of them as only one-letter abbreviations, A, T, C, and G. And the reason for that, see them listed here, is that there are 5 different nitrogenous bases: adenine, cytosine, guanine, thymine, found only in DNA, and uracil, found only in RNA. So because there's 5 different bases, that means there's technically 5 different nucleotides. But really, the only thing that's changing is the nitrogenous base. You can have A, C, G, T, or U. And there's a picture a little closer up. We'll talk more about these when we get to DNA in chapter, I think it's 7 later on this year. All right, let's take a look at the structure of nucleic acids and their polymer form. So again, once you get more and more of those monomers linked together, you now have a polymer. So DNA is linked together with a sugar phosphate backbone. So what that means is that the phosphate from one nucleotide links together with the sugar of another nucleotide. And you have that repeating pattern. Again, here's a phosphate of another nucleotide linked together with a sugar from another nucleotide. And here's the nucleotide, draw a square around it. This one happens to be an A, an adenine, and its phosphate is linked to the sugar of the nucleotide G, guanine, which is linked to the sugar of another nucleotide T, thymine. You'll notice in DNA, one of the big differences between DNA and RNA, shown here on the right, is that DNA is double-stranded. Here's a second backbone. There's a second set of nucleotides, a second strand, that's actually running in the opposite direction. We'll talk more about that as well later. But for right now, the basic structure is that second strand connects to the first one in the middle, kind of like rungs of a ladder. The bases pair up. A always pairs with T, C always pairs with G, and you get these alternating base pairs with hydrogen bonds linking them in the middle. So that's one of the big differences between DNA and RNA. We take a look at the RNA over here on the other side, and we have that sugar phosphate backbone. That's the same. We have the nucleotides are in the middle. What's different is that, number one, those nucleotides generally aren't hydrogen bonding to other nucleotides. Sometimes they do, but generally they aren't, so there's no double strand. And then, big difference, there's a U, uracil, in place of the T, thymine. It looks and behaves a lot like thymine, but it's a uracil, just slightly different than the thymine. Those are the big differences between DNA and RNA. All right, the formation of those nucleic acids. So how do we get nucleotides linked together? This should look pretty familiar, but watch some of the key terms that are thrown out there today. So unique among macromolecules, DNA actually has its own directions for replication. So that's pretty neat. DNA can replicate itself, and that process just, well, very descriptively named DNA replication. DNA replication occurs in what we call the S phase, the synthesis phase of the cell cycle. Again, we'll talk more about that later on in the year. RNA is actually a copy of DNA. We open up the DNA, read the DNA acts as a template to make an RNA. It's just a copy of the DNA. That process is called transcription. Transcription also takes place in the nucleus, but different than DNA replication. DNA replication is only really happening during S phase. Transcription happens pretty much all the time. You're constantly making new RNAs. And again, this is the part that hopefully is looking familiar to us. When the DNA nucleotides and RNA nucleotides, when they link together, here's that sugar phosphate backbone. That phosphate is connecting with the sugar, and our good old friend water comes off. So here we have our dehydration synthesis. Does it start to look familiar? We saw dehydration synthesis when we were linking together glucose molecules to make carbohydrates. We saw a dehydration synthesis when we linked together amino acids to make proteins. Here's another one. The OH is going to run away with this H. They form H2O. You've got water. You've got what's called a phosphodiester bond. That's the phosphate linking together the two sugars now. The function of nucleic acids. Hopefully you're aware, and if not, that's all right. That's what you're here for. DNA is the genetic material of life. So DNA codes for instructions that make most of life happen. RNA then is a very important copy of DNA that allows us to read the DNA, it allows us to read the instructions and use those instructions to make proteins. Kind of already talked about proteins. Here are those amino acids you'd see, all these little different colored circles. Those amino acids fold into proteins, and proteins do all the work of your cell. Why do you have the color hair that you have? Why do you have the color eyes that you have? It's all about your proteins, which comes from the RNA, which is just a copy of your DNA.